temperatures.
You're right, it doesn't look like he's actually eating the sponge bits so much as the sediment near the sponge. So Amanda's hoping that sponge is going to regrow from the part that's left behind. And actually maybe it will be able to do that because the crab seems to be leaving it alone at this point. Uh-oh. May have spoken too soon. Nope, there we go. Moving on.
Oh, and this
Didn't
crab
like what I was
has those little tube worms growing on it.
Wait, Amanda says there's a fish? Oh, there is a fish on the leg. Right at the bottom of the middle of the screen.
Yeah, if you could
And down
tilt down and zoom out just a tiny bit.
It's also missing one
There
of its
you
left
go.
legs.
Oh.
Oh.
Oh, wow.
Look at that.
That has got to be the smallest snailfish I've ever seen.
I was thinking snailfish, so yeah, you think so?
I think so. It's got the tiny little black to the tip of it.
What do you think, a 3 or 4?
I don't know. It's definitely a cute snailfish. Do they live attached to something? Do they kind of start out life like that? With like a little suction disc on its ventral side?
So Amanda's saying, or like a lumpfish? I don't know. Because it does look like it's really hanging on.
Yeah.
And it's so transparent or translucent that you can really see some of its organs.
Oh, it's inner workings.
It's very young.
Are we full zoom?
Yes.
Yeah. Wow.
Yeah, that's, I mean, all that's pretty small.
Yeah, Steve, did we get some beauty shots with the full crab with the fish in the view?
Yeah, we can come out and do some more of those.
Oh, zoom out.
Okay.
I think we got some wide shots when we just finished the sponge sampling, but we can
That
do
is.
Uh-huh.
a couple more. Lasers off.
Very nice.
Can we come in to see, to get the hose out of the way a little bit? Or is that going to cut it off?
Al, can you, right there. Can't really get the whole.
Yeah. All right.
The whole scene.
Yeah, have to crop the photo.
Yeah, that's as good as we're going to get. Okay.
Okay.
All right, got it.
Perfect example of a
Great.
All right, all the way out.
Thank you, Amanda, for noticing that. That's so cool.
Yeah, we're thinking that it's a snailfish, but it might not be.
Well, I'm looking up this idea of the lump sucker. I want to see how deep.
Yeah.
Because they have those modified pelvic fins.
Uh-huh. We did see full-grown snailfish earlier. We saw plenty of them.
Yep.
Science, do we want to continue to look around at any other features here or continue on our way?
I think we can continue on our way at a slow pace. Let's see, if we have to start coming up at 11. It's 10:05 now, so yeah, if we see one more, there might be time for one, maybe two more samples.
Roger.
Oh, Nav, did you get a target on that?
Oh, thanks. What is the number?
027.
I was too busy not putting in a ship move to give Bob some time to move. Okay.
All right, I'm going to hurry up then.
Well,
Before you do it.
Letting you go. Looks like you like to be about a half grid square away. that accurate? You like to be like this far out before
Oh,
that?
I go by,
Or
see
you go
that,
by that way?
see that,
Yeah.
I don't want to be outside of that.
Okay.
Then we're going to get yanked.
Yeah.
So. But since there's fishing gear and whatnot around here, we kind of want to stay.
Close.
Yeah.
Bridge enough.
Okay.
20 meters, 040.
So Justin is in the science chat, and he just added a link to a tweet from Ambari that snailfish attached to a crab. Snailfish lay eggs in gill cavities of crabs, and then they talk about tanner crabs and black snailfish, blacktail snailfish.
That's incredible.
Wow.
Yeah, it looks very similar. It's a good
So-
eye.
Interesting.
Says the person who knows nothing about fish.
Is that you?
No, I'm just looking
Oh.
at the Ambari photo.
Oh, yeah.
I was saying it looks very similar to the...
Yeah.
Yeah.
Well, that, I mean, snailfish, I think, was the first thing that both Jan and I thought of when we saw it. The little guy or gal that was there. So this is very interesting.
That's not a associated relationship that you immediately think of.
No.
No, not at all.
I think the story of...
There are some...
Oh, go ahead.
No, you go.
I was just going to mention there's stories of, you know, or not the stories, but the life cycle of a lot of smaller midwater fish involve a stage that is usually inside of a jelly bell, but never occurred to me
That's
that
what I was
crabs...
going to say.
Okay. Perfect. Jinx. But
great
crabs
minds.
are not what you normally think of as the home. Home sweet crab home.
At least not for a fish, maybe
Great.
a parasite.
Yeah. Yeah. Or crab
But
eggs. Yeah.
not for fish. Yeah.
Zero 40, right, Sam?
Yes.
Amanda, what time do you have to teach tomorrow? Oh.
Yep.
9 to 12.
Yeah. You can get... Want to see more interesting things?
So did the I was looking at the snailfish stuff. Did we leave that feature?
Thanks.
I think she's going to
Are we still on
I
it?
haven't
We're going to
had a
scoot
chance to
over.
look at high pack.
You can...
Oh.
You can kind of see things happening in the sonar up there, off to the
Okay.
off the left. Yeah.
Yep, just put in a ship move just for a little more north. We had moved originally away from the ledge because we were getting hit by a pretty strong current, but that seems to have died off here. So we'll inch back a little closer.
Shall I put sonar up on channel 3 or leave up the height?
I don't think so.
So we have under an hour left in Pioneer Canyon. And then we will be moving on our way, making our way down to Davidson Seamount and the Octopus's Gardens. Trying to beat the weather.
That's right. So we're gonna try
And I
and
think
count
Chad was
some
saying a
octopus really
3-hour
fast.
Yeah, a 3-hour descent
Wow.
from surface
Yeah.
to seafloor.
It's a lot of blue water. Yes.
That's a lot of blue water, and it's not something you can do quickly.
Will we be descending down to the location itself or get to the bottom and transit over to?
We're going to start at what we're calling the octocone. So that was a cone-shaped feature coming off the seafloor that we just barely had a chance to look around at the end of last year for about an hour, but we found additional octopus that were associated in cracks in the rock where there was warm venting water. So this is separate from the original location where we found the brooding octopus. So we're going to start there and
Jen,
try and survey that.
Is that what is being referred to as the octocone because it's a cone-shaped formation?
Yeah. Yes.
Cool.
Yeah, and so like I said, we only kind of looked around part of it, and so we're going to try to go up and circle around it and get a sense for how many octopus are there. We're not going to be doing quantitative transects.
Is that trash?
Yeah, right in the, looks like a can maybe, is that what you're looking at?
Yeah.
Do you see it?
A rectangular. It was just went out of view to the lower right. Well, it was rectangular.
It looked like something man-made.
Mm-hmm.
Yeah. Do you want to stop to zoom?
There it is.
Yeah, let's just take a quick peek.
Yeah.
Yeah, let's zoom. I think it's might be a. I think it's just a rock.
I don't know. It's not a
It's
can.
just so rectangular.
Yes,
Okay.
All right. Come wide. Optical illusion.
Looks like a wallet.
Yes, I was just thinking the same.
Well, then we should look inside, right?
Yeah, we'll find the owner. Return it.
Well,
Be good Samaritans.
Yeah, so back to the octocone. We're just going to try and get a sense of kind of an estimate of the number of octopus that may be living there. It's going to be a little challenging because based on what we saw last year, the cracks and crevices are wider and deeper, and the octopus were further down inside.
Yeah.
That's that right straight ahead. Yeah.
Zoom on the fish in the
Plastic
middle.
bottle.
Or something. Is it a fish bottle?
I can only zoom.
Yeah. Sports
A plastic
drink.
bottle. Hmm.
It's not very old. It's pretty clean.
Does it have a bunch of those worms inside it or something?
Yeah. A little habitat.
Alright, let's come wide.
That reminds me, Jan, I saw that and then I thought of plastic and then I thought of microplastics, which made me think of sediments. And then I thought we still have that one sediment core. I'm assuming this isn't going to be thick enough sediments
I don't
to
think
try.
this is thick enough. Yeah.
Yeah.
I think the sediment cores for this this dive and yesterday are it's kind of tough.
Jen, I wanted to clarify with you that sediment core we took today was about 3 or 4 centimeters. Did you want to preserve that in the core tube, or do you want to just put it in a bag?
Karina, actually, if you talk to Karina tomorrow, you can ask her.
Okay.
Specifically how she wants it.
Okay. We'll put it in the core tube in the freezer, I guess, overnight, and discuss that tomorrow.
Yeah, I think she did leave it written instructions.
What?
And
Okay.
I did put it in the Google folder.
All right. I'll take a look there.
And
I'm sorry, I can't quite remember
Yeah.
what she wanted to have done with the core samples. but she believes that there are some sleeves or containers that she left on board the last time she was on. She didn't give me any kind of containers or pouches for them.
Looks, I don't see nothing beyond it, but there's nothing on the Argus. See, like, looming seabed there.
So I don't know if this means that Justin has gone to bed or not, but he says, moving on, but forever changed by our adventures in Pioneer Canyon.
There has been quite a wide variety of habitats and aggregations of different kinds of species that we've seen over the past 2 dives. It's really been kind of exciting to be able to see what lives here.
Yeah, we had large expanses of really soft, flat sea pin habitat, and shallower as well as deeper.
Zoom in now.
We've had mixed fractured rock areas, and then these rubble rock areas, and some pretty steep
Alright, let's come wide.
ledges.
I'm not going to be able to look at these
Incredible
tanner
ledges, too.
crabs now without looking to see if there's a fish associated with them. It would be cool to see it twice.
Where's this?
It's going to be part of Amanda's students' project for the day. It's a science
Can we
writing
zoom out a
class.
bit more, Al?
But that's pretty much max wide.
Okay.
I can zoom out a little bit more, but it's very little, and it shows the bottle.
I think just because it's sort of uphill.
Yeah, yeah. No problem.
Is that a sablefish?
I think so. We've seen some, but they generally seem to be along the edges, like they're trying to get out of the light. This one doesn't seem to be quite as bothered by us as some of the others.
Mm-hmm.
Okay, let's zoom in. Like that.
Camera shot.
Alright, let's zoom out.
Science, we're about to get to near the end waypoint here. Do we kind of want to head due north from where we are? See what's up along near that end waypoint?
Give me a second here. I was looking up sablefish, and yes, that was a sablefish. I gotta find high pack.
I'm curious to see what's up at the top of some of this rock. If the current is stronger, maybe we'll see more corals and sponges if we go higher up.
Right.
Yeah, I
that-
would say just follow this ridge here.
Can do.
Yeah, this is a very nice area. I do want to take a look around, see if we can find Gary his rock pen.
Okay.
This I think is probably a pretty good habitat for it.
All right. Do you want to look around here or continue
Mm-hmm.
further down? As
When
in
you say
stop the
further,
ship and look around here, or continue moving up towards the ridge, or up the ridge?
stop the ship and let's look around here.
And the ship's stationary right
Are
now,
those
so
rock-
we've got some time.
Okay.
Are the rock pens always that color, that kind of darkish reddish purple color?
He's shown me one picture, and it was darkish reddish purple,
Yeah.
and so I think so. The description
Okay.
is that it's darkish reddish purple.
Okay.
And when I tried to look for some more images, they were all the same image, the same. All the different sites were using the same image.
I'm gonna reset Doppler.
Oh, yeah. Reset which one?
Reset the Doppler. I think it's all the it's fixing.
Oh, it's because, it's because Argus has a long max trail, I think. There we go. Now it's 2 minutes instead of 5. But it does look like Herc might be a little off.
It's not jiving with this.
Yeah.
Maybe.
Okay, give that a whirl.
So it was interesting. We went through that one section where there were more sponges than we'd been seeing. And now wait, what's straight ahead? Oh no, it's just a rock with Berzinga impact. Okay, for a second I thought it was something else.
So heading 350 should be towards the ridge.
Looks like another one of those sponges that we collected.
Hmm.
Yeah, Justin, I've got sleeping human upstairs, and he has not gotten much sleep the last 3 nights. Poor guy.
Yeah.
And tomorrow is our
Swing.
anniversary.
Argus cam.
Come on, corals and sponges. Where are they hiding? I think we're seeing more snails
check.
again than we've been seeing
Mm-hmm.
lower in that other location where we saw the corals and sponges. Now we're
20
seeing a lot
meters,
of
350.
Brazingid stars, anemones, thorny headfish. There's a deep sea sole or 2. Oh no, just one very large one there.
Just one deep sea sole.
Mm-hmm.
Say 2 or 3.
Hmm?
Sounded like he said 3 or 2.
No, we're good.
That's what I thought he said. Oh, okay.
Yeah, last year I got off of Nautilus and I went to the airport.
Pretty far in front of you, right?
Yeah, you're way out in front.
Yeah.
Miles.
Got on a plane, did a red eye to John's uncle's 100th birthday.
I'm gonna reset the DVL again.
That was what was happening
Something
a
funky's
year
going on.
ago. So now Uncle Al is 101.
That is a long life.
Yeah,
But
and
some of
he's
these,
still with it. Yeah, he's still with
That's
it. He
great.
plays cards. He loves his what are they? It's an East Coast New Jersey thing. It's an apple cider donut. Oh, he loves the apple cider donuts.
there's a coral
Yeah.
on a rock laying way over to the side.
Okay, my
All right,
V-Link
let's zoom in.
is starting to give out.
Nice. All
Every
right.
now and then my audio just goes mute on its own.
All right, let's combine.
So I was asking Eugene, some of the corals we've collected earlier in this cruise, they're going to be used for aging. Is that right?
One set is that keratosis.
So we can travel along this ridge here, not going over the side.
It's a bamboo coral.
Okay. Some of the deep sea corals can be very long-lived, right? Hundreds of years?
Yeah, yeah, a couple hundred years for some of them. It was a medium-sized one. We didn't take the largest. We didn't take the smallest. It was in an area that was extremely dense. I mean, it's definitely a forest of corals. And I think just recently on Chad's shift, they also picked up probably an Isidella skeleton, a dead one that had fallen over and was completely devoid of polyps. So and it's not as easy to age it. And Steve can talk to Karina about this tomorrow. But yeah, she has several. She has gotten from several different cruises skeletons, and I don't know. This is the first live one, full colony that I believe she's gotten, at least from sampling for her.
So, Jan, I'm noticing there are some Brazinghids just like under the lip, and I'm curious if we could look under there, because sometimes that's where you
Sure.
see like sponges or corals. Is that possible from a navigational standpoint? Ooh, look at that sponge too.
Yeah.
Sponge alert. Oh, but she thinks
Now,
might be
is
what we
that the
collected
taco?
earlier.
Do you think that's a taco sponge or the round? Not the round one. It's kind of yellowy.
All right, let's zoom.
I would hazard a guess that it's probably very similar or the same to the one we collected.
All right, let's zoom a
As
little
the
bit more.
taco.
Yeah, you can see how it's folded over a little bit.
Oh, it's got a crab in
It's got
there
that
too.
yellow tinge to it.
Yes, she's thinking it's the taco, but I love that it's got this crab living inside it.
Yeah, can we pan down just to get the sponge right in the middle of the frame?
Yeah.
Okay. And then some glamour shots.
Nice. Okay, those captures are good. You want to do lasers on that?
Jan, would you want the late? Oh, exactly what I was thinking. Oh, and
Yeah, that close.
are you noticing there's a little
close
shrimp
Mm-hmm.
looking out?
That's what I was really looking at. It's a little apartment building.
Exactly. Those look like little apartments for shrimp. I think that's pretty
Not
cool.
bad.
Yeah.
So Amanda's thinking that the texture looks a little different.
I don't think they're done looking at it.
Do you want to go back to that sponge? Sorry, I lost it with the current there.
I think that was pretty good imagery. I'm looking at science chat here. Okay, yeah, Amanda's saying it's just not different enough to warrant a collection. It's very small. So Jen, you were asking about going down and seeing what's underneath that ledge.
Yeah, just kind of
The ledge is
peeking
off to
under
the
there.
left.
Off to the left, yeah.
I think we could do that.
The thing is,
We
make
have
me think
about
there might
30
be some
minutes
good
left.
water flowing.
I think there's a strong enough current for at least for the corals, to really kind of hang out on top.
Okay.
It doesn't seem that silty, so they're in a pretty good spot. What's the depth range for desmophyllum?
It's quite broad. It's as shallow as 30 meters or less in some parts of the world, and as deep as a couple thousand sometimes.
Oh, okay. Because I have seen them on
Yeah.
ledges very similar to this kind of formation. So if we could take a look. Maybe just move along this overhang, little ledge.
I don't know how much room we have with Argus. Oh yeah, we're pretty close.
What is desmophyllum?
It's a stony little cup coral.
Oh, okay.
It's very fragile. It's very hard to collect.
Okay, I see it in the spreadsheet, but there's no image.
Oh, it's not? Oh, okay. Is it related to Lophelia, or is it just like one of the stony corals?
So it's a bit of a complex history. So Lophelia is a colonial coral species, and Desmophyllum dianthus has been largely considered and is a solitary species. But until a few years ago, there was a revision done from some findings looking at the genomic data from Lophelia pertusa that found that perhaps Lophelia is more similar to the solitary species Desmophyllum, and so the name was recently revised for Lophelia into the genus Desmophyllum, and that's where things stand today.
Oh, wow.
but there's some disagreement about that. So it's in flux or under investigation by multiple parties right now.
And what size are we looking for?
Small.
How?
10 centimeters.
10 centimeters. Yeah, I was going to say half the width of the lasers.
Okay.
I think smaller than 10 centimeters, right?
There's a white sponge on in the upper portion of the screen.
And is that a snailfish behind
I
it?
don't get it closer.
Yeah,
I
yeah.
was
There's
You
not
get
curious.
light
one
sky,
head of
you
some
get
type.
light.
Oops. Oh.
I would just plant tilt up.
The ship's stationary,
Nope,
so we can
he's
look
going to
around.
come out. Yes, snailfish.
Sclerothamnopsis.
So that was the first one then that
Zoom.
we collected.
Mm-hmm.
Okay.
Mm-hmm. Yeah.
So many little cracks and crevices on this rock. All sorts of things could be living here.
Yeah. We've seen layers like this and just rows and rows and rows of different species of rockfish living in there. So it was kind of like a little condo.
Thanks for the zoom.
All right, let's come back wide.
There's a couple things here that give me some concern as to why we're not seeing those stony corals. Namely, the oxygen is quite low here still. We've been hovering between 3 and 4 micromolar for almost the entire dive. And I'm not too sure about this substrate too. It's kind of some kind of mudstone, and I don't know how stable it is over the life of a hard coral.
Yeah. There's a lot of slumping and earthquakes around here, so things do get rattled. Even though this is on the Pacific plate. And usually the movement's on the North American plate.
So Steve, are you thinking we have more luck on the top of this than on the side of it?
I think it's going to be similar in both places. I think it's good we got a look at the ledge. The current isn't as strong as I expected for this little channel here. It also could be a factor why we're not seeing as many corals. It's somewhat of a stagnant current.
Science, it's 1040, and looking at our current depth of 800 meters with an ascent rate of approximately 18 meters a minute, we've got about 44 minutes to ascend. So I think we've got about 40-ish minutes before we need to head up to the surface. How would you like to spend that time?
Okay. I think just going along this ledge right here, and once it ends, we'll pop back up to the top. Keep moving in this direction.
Yeah, there's a lot of sponge right there.
Okay. We are currently about to pass the end waypoint that was on HIPAC, so we can just kind of continue north with the hopes of following this ridge. Okay.
Yes.
Copy that.
Yes.
There's a good example of the long spine thorny head in the foreground. As we pass over, you can see that longer spine, third spine.
Yeah, what is that framework in the back? Kelp?
like it.
Line?
Oh, I hope so. I don't think so, though. I think that's line.
Oh.
I think that's rope. Something really stiff. And it looks like right in front, just kind of 2 o'clock from that white sponge, is something that looks like line. Or maybe tilt, but
The ship's holding position if you want to take a zoom.
Yeah, just
Yep.
this the rock's kind of in my way.
You could pan over there with the camera too.
Yeah. Just right on the edge of this ledge.
what is that crab doing? It's like rearing up.
Well, the one across from it also seems to have
Okay.
I don't know if you're gonna be able to.
Oh.
You're not gonna be able to
Reaching
pan all
up.
the way over there. You could zoom that maybe. See.
Alright, Al.
I wonder if they, see each other in a different, they don't usually
it's
see each
definitely
other, right? There's no
line.
light down here.
Yeah, let's zoom back out a little bit. Yeah.
Net. Sufficient net.
Could it be
Yeah,
a
we can zoom in.
kind of a cage, too?
Are you looking at that line? Is that what you're looking for specifically?
Kind of both of these.
Just the one.
Alright. Zoom back out.
Can we get a close-up of the 2 crabs up top?
The top? Yeah.
Up top, yeah. They look like they're about to duel.
Don't get over where that is.
Yeah, we don't want to get near the.
I don't want to get over.
You don't want to get over.
No,
where
we
the
don't
line
want to get over.
Yeah. Just so we make sure we budget enough time here, I have in my notes a request for a shallow Niskin sample with the last one we have somewhere within the 50 to 100 meter range. Is that consistent with what you all have watch leads? All right, Al, let's zoom in on those crabs.
We took the 50-meter one earlier when we sampled the coral for aging.
Okay.
And we only have one left. So let's do one 700 meters
Okay, let's come wide.
above seafloor. So it's going to be closer to the surface.
Yeah.
Okay, copy that. Is that in a 10, 15 minute? Less than that?
Oh, yeah, that should be quick.
Okay. 5, 10 minutes.
Oh.
Aim for a like 11:05-ish then. Starting to head up.
Yeah, I mean, it should just be stop, ascent, pop the line, and
Okay,
yeah,
perfect.
continue on.
11:10. That's the range.
Yeah. 11:07 Q, really cool
Yeah.
thing.
Right? That's when the whales show up.
Oh. Well, earlier they were talking about the depths at which marine mammals can dive, and these Dover sole they were seeing, and that northern elephant seals can dive this deep. And I was thinking, with all this Dover sole there, it would be amazing if this elephant seal had come down to that area. So, if we had something amazing like that happen right as we're about to leave, that would be something to see for sure.
Yeah, unfortunately there's a nice little coral on the side. The elephant seals that dive that deep are older, and they migrate out to basically the middle of the Pacific, take a little vacation either in the Gulf of Alaska or Hawaii, depending if you're male or female, and feed on just very, very deep fish.
Mm. So they're not here right now, is what you're saying?
No. And the ones that would dive around here would be young, and most of them will eat ratfish, nyctophids.
Yeah, but you would think
I
like
think
a gravid Dover sole would be quite a meal.
Harbor seals and sea lions, they'll grab a Dover sole.
Okay.
I don't think that they won't dive that deep, but
The
fish are
shallow
shallower.
ones.
Yeah. There's some algae. Did Taylor Ann get enough of her algae?
I think she's all set, yeah.
Okay.
She didn't make any more requests. She's got algae, kelp from yesterday, kelp from today.
I think that there's a crab possibly dining on that piece of kelp.
I love that thorny head facing us, just peeking out from the rock.
That kelp's a good reminder, though, that quite a bit of this community of animals are fed by productivity that's made in the portions of the ocean that get sunlight by photosynthesis, and then over time it floats out or dies and sinks down to the seafloor here. Some of the marine snow that's being caught by the filter feeders, and some of it is being eaten off the seafloor by the scavengers. Supports all these animals that we're seeing out here.
How do commercial fishermen fish for tanner crabs? Do they use like crab pots, traps?
I don't know, but that would be my assumption.
Yeah.
Yep.
Oh, there's a whelk egg tower back there.
Do we want to check it out? There's a couple of
Is there anywhere else that we want to
them. A great potato.
I think just keep going in this direction. This is pretty habitat. A lot of Pioneer Canyon is this fractured rock. Some areas have pretty large boulders just comprised of fractured rock. It still hasn't broken apart. Is that a scallop? Oh,
Jan, when we're exploring like this, do you prefer the lasers on or off? I just noticed they're not on.
Can we do a snap zoom in there?
oh, on. Yeah.
Is that a scallop?
I think I'm betting
That's what I was
anemone,
wondering.
but
Huh.
Yeah, I don't think scallops,
Too deep?
at least that large of a
what's the
scallop.
name?
Acesta. Acesta? Is that how it's pronounced? Acesta.
Now?
A-C-E-S-T-A-L.
Yeah, ready for zoom. Okay.
oh, yeah.
Uh-oh. A. Cesta.
Oh, look at that.
Wow. I don't think I've ever seen one this deep.
Good eyes, whoever spotted that. Wow.
Sorry, Steve.
Yeah.
We should have bet some cookies or something on that one.
All right, let's come wide.
I'll stick to corals. Anytime I try to ID things with backbones or things with shells or rocks, I always fail.
Oh no.
Yeah. Usually fail. Sometimes I get lucky.
All right, come back in. Are we good with shots on this?
Yep.
All right, let's come wide.
So Justin just sent us a link to an article from the Burke Museum about snailfish eggs in the gill cavities of king crabs, and they're snailfish laying their eggs in these king crabs. So pretty interesting. Different species of crab, different species of snailfish, but looks like we might have the analog down here with the black-tailed snailfish and the tanner crab.
And Amanda sent us a link to an Mbari website for Acesta. Sphony. Sphony? No, not Sphony. Sphony. Sphony.
Is that the scallop?
That's the scallop, yeah.
Huh.
What is that? Yeah.
Big tower with a snail on top of it.
Oh,
Yeah.
that's great. That's a good photo. I guess we already got a photo like that earlier, though. Okay.
But we didn't get a photo of this one.
Good point.
Say that to all my bird students. Oh, we've already seen that one. No, but you haven't seen this one.
So I'm looking at the Acesta from the Mbari Deep Sea Guide, and the sightings actually go a lot deeper than our depth right now of about 800 meters. They've seen them all the way down to 2,400 meters. So we're actually on the shallower end of the distribution of that scallop.
Open more. See if I can hold it.
Oh, okay. It's not a scallop.
Oh, it's not?
Oh. It's Limmidae, the family Limmidae, the file shells. There you have it.
Mm.
The ship's holding here so we can look around as much as we like.
Okay.
And now we have some photos of this one.
Yep. Any more zoom on that, or are we good?
Yeah, I can zoom.
Let me center it a little bit.
Yeah, that's what I was waiting on. Oh.
And might have lost it with that.
Oh, and I was wondering what was on this snail shell, but it's a shrimp.
I'm coming
Let's
out all
just
the way.
come wide, yeah.
Oh. I'm surprised that the crabs don't eat up the eggs. Must be something not tasty about them.
Yeah, I don't know. They appear to be kind of like they have a thick coating, but never having seen them up close, it's hard to say.
So Justin's been researching the snail, different crab species, but does name black-tailed snailfish. Surprisingly, the results yielded evidence of 4 species of Caraprotus, Melanurus.
Oh, I think it's Caraproctus.
Terrible at Latin.
Caraproctus.
Caraproctus, Melanurus, Pharcellus,
I think that's right.
and Simus. Oh, and Coletti.
Coletti, yeah. Those are the different snailfish species, I think.
Oh, can we stop? Let me just take a look at that coral.
Which coral?
Just went out of view to
It's
the right.
3 o'clock to the right. Just went out of frame.
Oh yeah, I see it.
Oh, it just might be a really skinny Paragorgia. I'm going to take a look at the list here.
All right, now let's zoom in a little bit.
Can we go more in?
Just seems like the axis is
I can tell.
kind of more
All right, good
circular
capture on that.
spiral.
Let's come wide. Are we good on that coral?
Yep, I think so.
Yeah, if we could hold here for just a second.
Okay.
I'm checking the list here.
Yeah, while we're waiting, there's a sponge to the up and right of that coral under a ledge. Do you see the sponge? You go from the coral and go
Yep.
like up and to the right.
It almost looks like a great potato.
I don't think so, but
It
I
does look like a group.
it would be great if it was.
Okay. Yes, I'm done with the coral. Thank you, guys. We can go look at the gray potato.
I don't think it's gray
Is that
enough.
what you were looking at? The little face sponge?
I think so, yeah.
Okay.
Right in there in the center.
Now in my center.
Well, now it's in my center, but I think we're a little behind you.
Let me know when you're ready,
You
Bob.
zoom in.
It's the end of my lens. Oop, zooming out.
Okay, that's good enough on the zoom, she said, to be able to see that sponge. Thank you.
You can